This is the third lecture on Chapter 11, discussing neuropeptides, the classification, synthesis, and colocalization with classical neurotransmitters. Actually from the PowerPoint slide presentation, or the PDF file. It begins with slide number 26. However, we'll view, other than this title slide, one additional slide to provide the overview of the entire chapter, in essence, as it relates to the discussion in these remaining few slides. This slide is from an earlier part in this PowerPoint presentation. It is from figure 11.1 in the textbook, and it was comparing the life cycle, if you will, of the classical transmitter with the neuropeptide, and we have discussed the distinctions in synthesis and transport to the terminal to the site of release for the classical neurotransmitter versus the neuropeptide, and stated that the basic distinctions are that the peptides depend directly on gene expression for the production of the transmitter itself. As well, there is no synthesis at the vesicle, so the readily releasable pool available in the nerve terminal for release when an action potential arrives is completely dependent directly on gene expression back at the soma. Let's now focus on the life cycle of a neurotransmitter at the terminal, and then on occasion compare that with that of a peptide. They have very similar properties, as we will see. Thus, then, this slide is showing us the life cycle of a classical transmitter. At the first point, for transmitters, again, recall that synthesis can occur at the terminal. The biosynthetic enzymes are present either in the axoplasm or in the vesicle fluid, the intravesicular fluid. Thus, there is some type of a precursor, for example, an amino acid with a specific transport protein on the the exoplasmic membrane, or the axolemma. The precursor is acted on by these biosynthetic enzymes, as we said, converting, for example, X to Y. Y has to be loaded into the vesicle. So there is a specific vesicular transport protein for this Y. It may be the final transmitter that would be released, or a precursor, and then the final production or synthesis will occur inside the vesicle. We know there's a process of moving these vesicles toward the presynaptic membrane, leading up to the exocytotic release into the synaptic cleft, biological effect at the receptors, and there's potential for, and then there's removal from the synapse, reuptake presynaptically, postsynaptically, or by nearby glial cells or other cells. This slide reminds us that what is really important in this type of neurocrine signaling for a neurotransmitter, as well as for a neuromodulator that might diffuse into this area, or if a transmitter diffuses out and is going to act as a neuromodulator, it is the interstitial, in this case synaptic, concentration of the extracellular chemical messenger that is extremely important. So the key components of sustaining the synaptic concentration, let's focus on that for the moment, are release, which is dependent upon many processes that include, of course, synthesis of the chemical messenger. So release, or the input, and then removal. Here we see reuptake as one mechanism, and of course there is certainly enzymatic degradation for the peptides, which is considered to be the primary mechanism for removal of peptides from the interstitial fluid compartment of the brain, especially the synapse. I'm just going to set my pointer. So we have steps 1 through 6 shown here, and we highlight them in this slide. One was the uptake of the neurotransmitter precursor into the presynaptic element, because synthesis can occur at the terminal. This would not be the case for the neuropeptides. They would have to be synthesized at the level of the soma due to gene expression, transcription, translation, post-translational processing, packaging, transport, and delivery finally to the terminal for contributing to the readily releasable pool. No vesicular transport from axoplasm into the vesicle. Otherwise, after synthesis, all the distinctions we mentioned, and the accumulation and storage of the neurotransmitter into the vesicles that would occur via the ER transport vesicles. More importantly, the Golgi apparatus, of course, that we know that produces the vesicles. So big distinctions between the classical neurotransmitters like monoamines, GABA, glutamate, so on and so forth, versus the neuropeptides. Vesicular release or exocytosis of the neurotransmitter, that is shared, something they have in common. Interaction of the neurotransmitter with its receptors. We have no problem envisioning that for neuropeptides as well as classical neurotransmitters. Inactivation of signaling through one or both of the following. Uptake of the neurotransmitter by transporter systems. This is not well established for neuropeptides. However, there are some evidence that this does occur. It's not completely understood, and it is certainly not the same as any evidence that does exist indicates something different happening than what we see for the classical neurotransmitters. Certainly enzymatic degradation in the synaptic space is the prime method of removal of the neuropeptides as biologically active molecules from the interstitial space, synaptic space for neurotransmitter, peptides, neuropeptides acting as neuroregulators through a neurotransmission mechanism, and as well for neuromodulation if it's acting non-synaptically. The reuptake for the classic neurotransmitters, we're not to confuse that cytosolic uptake or axoplasmic uptake, shown here, presynaptically, postsynaptically, and extrasynaptically, with the vesicular accumulation. So classical neurotransmitters have in their axon terminals vesicular transport proteins. For example, VMAT, vesicular monoamine transport protein, is on the vesicles and will transport the monoamines that were taken back up into the cytoplasm into the vesicles, recharging them so that they can be recycled for re-release. And that is in conjunction with and concomitant with neurotransmitter synthesis and charging of the vesicles as well. So they're both transport systems, but with different functions and mechanisms. Sometimes agents will act on both, such as amphetamine will block the dopamine transport, the DAT, as it is called, DA for dopamine and T for transporter. So amphetamine will act here to inhibit that, as well as the VMAT on the vesicles and inhibit that. Dopamine concentrations increase in the axoplasm, and ultimately the DAT acts in the reverse direction, and that's how amphetamine ultimately will increase synaptic concentration of dopamine by reverse transport of dopamine from the axoplasm, and it becomes independent of action potentials, because we know, as we'll see, action potentials are required for exocytotic release. So it's a good segue into talking about the exocytosis. If you don't put transmitter into the vesicles, you can have all the action potentials you want. You don't get any release into the synapse, the interstitial fluid. If you reverse the direction of transport of these reuptake molecules and the synthesized transmitter is present in the axoplasm, then that will dump the transmitter into the synapse independent of action potentials. So a little additional information there. And as I said, a segue into this slide showing us the action potential coming down the exoplasmic membrane, the axolemma, the axon membrane, the axolemma, changing the voltage of the membrane, depolarizing it so the voltage-sensitive calcium channels open, localized influx of calcium, causing a localized intracellular increase in calcium, free calcium concentrations. This triggers the docking of the vesicles, and then an energy-dependent, ATP-dependent mechanism for the fusion, and then there is diffusion down the concentration gradient, obviously higher concentration in the vesicle than in the synapse, so diffusion is down the concentration gradient. This is a high magnification electron micrograph. The scale bar here is 200 nanometers, and this is the presynaptic membrane and the postsynaptic membrane, and we can see what's called the synaptic web, some electron dense material in the synaptic cleft itself. And we can see a variety of things, vesicles scattered throughout. We can almost see the lipid bilayer clearly in all of these. So some have not docked yet, others have docked. We don't know exactly what plane of section we're seeing here. It could be off to the side. For example, if you were looking in this direction and you had a plane of section somewhere through here, it might look like this. So we don't really know what that is. This is a classical omega figure opened for transmitter diffusion out into the synaptic cleft. Next, then, we can consider Dale's principle, which basically stated is one neuron, one transmitter. It's a little more sophisticated than this. Basically, the caveat is that for any one synaptic interaction with a target cell, one transmitter is being used, and in essence, we probably should place Dale's principle into historic perspective and virtually abandon it. Is this principle violated by colocalization of multiple transmitters in single neurons? In my opinion, that's a consideration that is academic only. We just have to know the details. Is there a colocalization? What's the primary transmitter? And what is the co-transmitter? When are they released? Under what conditions? What do they do when they're released singly or together? There's much that we know, and still much more that we need to know and understand about this type of complex signaling at a neurocrine functional system. So in this slide, a figure from the text, we see the presynaptic terminal, the postsynaptic membrane, the small vesicle shown in here with the solid dot contain only serotonin or 5-hydroxytryptamine, HT for hydroxytryptamine. Substance P alone is the larger one, solid, dense core vesicles, and if they're colocalized, it's large with half clear and half dark, half filled. And of course, the serotonin is the primary transmitter at this signal. The peptides are almost always secondary transmitter if they are colocalized with what we consider as the classical neurotransmitters. In the lower panel, we're seeing a primitive neuron developing into either an endocrine cell or a neurosecretory cell or a neuron. So there's this common descent of lineage in many cases, and this leads to a discussion of the APUD, the APD system, as we will see. And it is conceptual as far as general research or science is concerned, but clinically there is some relevance to it because primarily with pathologies, of course. Cells with common embryological origins have typical and common tendencies in gene expression, behavior, and therefore pathological symptomology. But we'll talk about that more in a moment. Let's first continue with the discussion of the colocalization. I'll give you the source of this information. It's from the ACNP, the American Colleges of Neuropsychopharmacology. They have a free text at this website. and this one was on the colocalization of peptides. So peptides coexisting with norepinephrine are reflected here. They're projections in the central nervous system. It's a parasagittal view from the side, basically cutting from nose to tail and looking from either the right or the left side. And it depicts the coexistence of galanin, GAL, or neuropeptide Y, NPY, and the norepinephrine, or NE, in the locus coeruleus and the A1 cell group. They're numbered. This is the first adrenergic cell group down here in the lower brain stem, the medulla, just before it meets and joins with the spinal cord. There's the ascending, upward, higher in the CNS, and descending projections, obviously, down into the spinal cord. So there's a solid green set of lines that indicate the galanin projections, and dashed red lines indicate the NPY projections. The line thickness indicates the percentage of peptidergic neurons projecting to the terminal region, so how heavy of a projection or pathway is it. So, for example, locus coeruleus into the hypothalamus, where the paraventricular nucleus is located, is a very heavy projection. Locus coeruleus is carrying information from the sensory nervous system, primarily the somatic sensory and visceral as well. So changes in the body due to the reactions to the environment or directly from the environment carried through here, and then the central norepi system contributes to the centrally mediated components of the fight or flight response, right? Descending noradrenergics affecting the autonomic nervous system, of course. So excuse me just a moment. So some galanin projecting, galanin is, some of the locus coeruleus norepinephrine neurons are colocalized with galanin and are projecting out of the locus coeruleus. And this heavy is 30% projection, ascending, and the lighter one is 3 to 14% projecting. For the neuropeptide Y, the dashed, we see again a heavy ascending projection from both locus coeruleus and the A1 region. This A1 region is receiving a lot of chemosensory and blood pressure type of information, taking it to the hypothalamus specifically to integrate with neuroendocrine responses to changes in the cardiovascular system predominantly, but there are other related functions. So with hemorrhage, this norepinephrine is very important for regulating antidiuretic hormone release. So we see a lot of neuropeptide Y, and as well from the locus coeruleus up into the thalamus, the hippocampus up here, and the throughout the cerebral cortex. So the colocalization of the peptides with the classical neurotransmitters is extremely important, and obviously to a large degree beyond the minimal scope of our course. But we are definitely getting a greater appreciation for that. So there are other topics in Chapter 11 that basically there will be details of which in the upcoming lecture materials. So the neuropeptide receptors and second messenger systems will be a focus of separate chapters. The important, and we'll see it again and again throughout the semester, importance of neuropeptide agonists and antagonists. I have a table to comment on that. Identification and localization of neuropeptide target cells. We'll discuss this with the methods lectures. And neuropeptide activation of second messenger systems, of course, is a key part of what we will discuss for hormone action in general, as well as the neuropeptides acting as neurotransmitters or neuromodulators. So this is a table again from the ACNP site. on the first non-peptide antagonist. So this is the point that I wanted to make because it is of great importance clinically and, of course, economically if you're investing in the companies that develop one that would have wide impact on clinical treatment of some disease or pathology. These are used more so for research, but there is some, obviously, if it is a wide variety of drug companies that are developing these, there is hope of clinical utilization and cashing in on that, because simply selling them to researchers doesn't make quite as much money as having a clinical application. So for cholecystokinin A receptor, the code name is L364718 by MSD. I don't recognize this company's name. Eli Lilly, I certainly recognize. The LY will always tell you that it's an Eli Lilly compound 219057. So you might hear scientists using this terminology, and they have all of these memorized, but typically within their own field, not too much crossing over from one to the other. A lot of numbers to memorize. These are important if you want to do a literature search. Put these in, and you should get a great deal of information on these, the utilization and the research completed on these compounds. Cholecystokinin B receptor, we see the three of these, Park Davis getting in there, and the neurokinin 1, neurokinin 2 receptors, and neurotensin receptors. So there's a wide variety of these: Pfizer, Roan, Polenc, Sterling Winthrop, Sanofi. So a lot of different drug companies involved in producing these. And there are many new non-peptide agonists and antagonists. So why non-peptide? They can be orally active. They're more likely to be orally active than the peptide or an analog of the peptide itself. You might make an analog of a peptide that will extend its half-life so that it will be biologically active for a longer period of time, which is important clinically, but you would have to administer it intravenously. If you wanted it to act at the brain, it probably wouldn't pass the blood-brain barrier in concentrations high enough to really have any major effect. Some of these can be designed so that the lipophilicity is modified. You decrease lipophilicity, you can limit it to peripheral effects. You increase lipophilicity, and you can target central effects as well as the peripheral effects. It's somewhat difficult to make it selective for crossing the blood-brain barrier but not having an effect in the periphery. But you can differentiate peripheral effect, isolate peripheral effect simply by decreasing lipophilicity so that it will not cross significantly across the blood-brain barrier. Now just quickly a discussion of the APD cells. These are amine precursor uptake and decarboxylation cells. These cells are derived from primitive cells in the neural crest. We'll take a look at this so you can understand it in a moment. During ontogeny, they migrate to the foregut and specific locations of the neuroendocrine system. Properly located, these cells form part of the normal hormone-producing tissues of the neuroendocrine system. If misdirected, these cells may become hyperplastic, adenomatous, adenomatous, let me get that pronounced correctly, or malignant. So you can cross-reference that with apodoma. This is the source of this. It's Webster's online dictionary. So pretty common type of information. So these common embryological origins are illustrated on this slide. This is a dorsal view of the human embryo within close to the end of the 3rd week, so that the 3 embryonic germ cell layers are established. Ectoderm, the red here is the mesoderm, and the yellow is the endoderm. The cross section here is showing it better. Ectoderm on the surface, mesoderm in the middle, and endoderm on the innermost portion. So this would be the back side of the embryo and the front side of the embryo. We see the neural plate being stimulated, these ectodermal cells stimulated by this structure called the notochord that develops, and these will become the nervous system, and we want to follow them as they get deeper and deeper and become the neural folds as shown in this slide into the 4th week of the human embryo. So first, and it starts at one location and then becomes complete in the mid-axis of the embryo first, and as you progress away, so as we look at a series of cross-sections through this human embryo, we see different stages of development. So the neural ectoderm was stimulated by the notochord, and it continues to develop. We see the neural plate here deepens to form the neural groove, and alongside this lighter blue is showing the neural crest cells. So as we follow this, get deeper and deeper, forming the neural groove, the neural crests approach each other, get closer and closer. They begin to develop and differentiate as the tube is almost formed, the neural tube is formed, and these neural crest cells form this little inverted U-shaped structure dorsal to it or toward the back. This is now all just going to become skin up here. And the neural crest cells take place on the dorsal, lateral, on the right and left sides of the neural tube, going to differentiate into the sensory neurons, but also all peripheral neurons, like the sympathetic neurons, the postganglionic sympathetic neurons and parasympathetic neurons, but all the sensory neurons. But some of them go off and do special things, such as ending up in the gastrointestinal system, differentiating into what are called enteroendocrine cells or enterochromaffin cells that'll synthesize and secrete a variety of hormones, just epithelial cells on the surface of the lining of the gut tube. Serotonin is a major hormone synthesized by some of these enterochromaffin cells. But let's use the adrenal medulla and the sympathetic neurons as an example in following some of these neural crest cells. So here's the surface ectodermal or epithelial cells at their junction with the neural plate. These neural crest cells break off or become migratory and move away. The crest will come up on either side and fuse to form the neural plate. This is a cross-section of a mouse embryo showing the neural tube and the notochord with the scanning electron micrograph. And the neural crest cells are right in this area here. They're highlighted in red so we can see them. They're beginning to migrate into this dorsolateral position, but some of them continue to move on through the mesodermal layer to populate the sympathetic postganglionic neurons throughout the body where they're located, sympathetic trunk, the sympathetic chain, in the wall of the gut tube, and so on and so forth. So the multipotent neural crest cell will continue its development to become a bipotent precursor cell that, if in the right location and affected by fibroblast growth factor 2, will begin to express the nerve growth factor receptor gene, therefore become nerve growth factor competent cell, and then in the presence of nerve growth factor differentiate into a full-blown neuron, a sympathetic neuron. If, however, this bipotent precursor cell ends up where the adrenal medulla is going to be synthesized or somewhere else aberrantly and become exposed to the appropriate levels of glucocorticoids, such as coming from the adrenal cortex, developing in that area, then certain genes will be expressed, and this will become a chromaffin precursor cell. With the continued exposure to the glucocorticoids, it'll become a chromaffin cell that, of course, synthesizes and secretes adrenaline, similar to, well, a bit different. There's one more enzyme that is expressed. This sympathetic neuron, postganglionic, will synthesize and release predominantly norepinephrine, will not express the gene for the final enzyme to convert norepinephrine to epinephrine. We'll study the details of that later. The adrenal medullary chromaffin cell will express that gene for that final enzyme to convert the norepinephrine to epinephrine, and epinephrine will be the dominant molecule hormone, in this case secreted by this adrenal medullary chromaffin cell. So other peptides are synthesized and released by these cells as well. But still, monoamine being released by this cell, monoamine being released by this cell, the serotonin being released by the cells in the gut lining, as I'd mentioned. So they have to have a way to uptake the amine precursor and ultimately to decarboxylate that so that it produces one of the biologically active monoamine molecules that can act either as a neurotransmitter or a hormone. So therefore the amine precursor uptake and decarboxylation system. It's a bit esoteric, but in the textbook, relevant, allows us to think about differential development of cells, impacts of different hormones on gene expression that are important for lineages, different lineages for cell differentiation, to the terminal differentiation for the functional cell, and that they can have commonalities. This ends our lecture.